Hello and welcome everyone. My name is Tate. I am a PhD student at the University of Adelaide and am part of the larger CRC for honeybee products. Before I continue, I'd like to acknowledge the traditional owners of the land on which we meet today. I would like to also pay my respects to elders past, present and future. My PhD is part of a larger project within the CRC for honeybee products. That project aims to develop a bioactive Leptospermum breeding program for South Australia. The larger project is divided into three key parts: understanding the genetic diversity to support selection of superior lines, development of robust propagation methods, and discovering how environment influences plant growth, flowering and DHA production. Today, I will be talking to you about the interesting world of Leptospermum propagation. To give an outline of today's talk, firstly, I will give you some background on the importance of bioactive honey and how Leptospermum is an important source. I will then present the pros and cons of propagating Leptospermum through the three most common methods. I will finally give you an insight into the propagation work I have been conducting as part of my PhD. For the background, all honey can be considered bioactive to some degree. Bioactive refers to its ability to have a biological effect in certain circumstances. This effect could be antibacterial, anti-inflammatory, or antioxidant. However, some honeys are much better and have greater bioactive capacity than others. These honeys provide an opportunity to treat antibiotic-resistant bacteria, as well as provide support for the medical and nutraceutical industries. The antibacterial qualities of honey stem from a unique set of chemical properties and the interaction they have with living organisms. These relate to the pH, the sugar concentration, and production of hydrogen peroxide upon dilution, all of which reduce microbial growth for one reason or another. However, there is a final property that not all honey has. This is its ability to produce antibacterial effects upon continued dilution. This is often referred to as the non-peroxide activity. The most famous of these non-peroxide bioactive honeys are Manuka, which is produced from Leptospermum scoparium, and jellybush honey, which is produced from Leptospermum polygallifolium. The additional bioactive strength of these honeys is correlated to the concentration of methylglyoxal, or MgO, in the honey. Methylglyoxal is derived from dihydroxyacetone, or DHA, in the nectar of some Leptospermum species. There are 83 species of Leptospermum that are native to Australia across a range of environments. Of these species, many have been found to contain DHA in the nectar. At least 20 species are recorded producing concentrations of DHA that are greater than Leptospermum scoparium. The broader questions of our project are very much focused on which species might be best suited to plantation production of high-grade bioactive medicinal honey in South Australia. Influencing our thoughts on species selection is Manuka is equal to Leptospermum scoparium, but is that the best species for South Australia? In South Australia, we have 5 native species of Leptospermum and 1 introduced species. Of these, there are 3 that have the potential to be equally as good, if not better, than Leptospermum scoparium. A benefit to the local species is they already grow in a range of environments in South Australia, potentially having adaptations to the local environment that scoparium may not have. These could be hybridized to produce attractive and adaptable plants with bioactive nectar. A quick review of the availability of plants in South Australia show that scoparium is mainly sold here as an ornamental plant, as we can see here in the State Flora Catalogue. This catalogue also provides good information on the growing conditions for Leptospermum continentale and Leptospermum lanigerum. So now I'm going to give you a brief rundown on seed propagation. Leptospermum can be propagated from seed. This method is usually cheap, simple, and promotes diversity within a population. It is also the current practice for mass production of most Leptospermum species across Australia. However, DHA varies within populations, and it is expected that it may vary within seedling grown populations. As with the DHA levels, a number of other traits relating to flowering and growth will likely also vary when seeds are grown into plants for plantation. There can also be issues germinating Leptospermum seed. Leptospermum from WA especially require pre-treatment to encourage germination. So an alternative to seed propagation is clonal propagation. This can be done with tissue culture or using cuttings. Clonal propagation from tissue culture is one alternative to growing from seed. It is useful for difficult to propagate plants, provides an option for fast multiplication of material, and can maintain desired traits over generations. However, it is a complex process that requires specialist skills and equipment to conduct. It is also labour intensive, and plantlets require special care in the process from taking them from the lab into the glasshouse environment and then again into the nursery. An alternative to tissue culture for Leptospermum is cutting propagation. Propagation from cuttings provides material that maintains desired traits across successive generations. The process of production from cuttings is simple, scalable, and uses readily available materials and equipment. It does have its pitfalls though. It is more labour intensive than seed and requires a level of control over the environment. As a result, plants produced are more expensive when compared to seedlings. There is also a lack of detailed methodology for propagation and subsequent transplant survival in published literature. To overcome this lack of published literature, I have been investigating the development of a methodology for Leptospermum propagation as part of my PhD. The decision was made to investigate cutting propagation methods on Leptospermum scapparium first, as it is responsible for Manuka honey. As part of this, I investigated how genotype, auxin application, plant material age, and watering in affected propagation and transplant survival. After planting 4,000 semi-hardwood cuttings and 6 months of time, it was found that auxin application and watering in cuttings after planting had the most significant effect on plant production. To maximize scapparium plant production, 5 to 10 centimetre cuttings should be treated with 3 grams per litre IBA. They should be watered in and placed into a humid environment. After about 6 to 7 weeks, cuttings can be transplanted. At this time, the strike rate, or the amount of cuttings that produced roots, was approximately 80% of those that were planted. Over the coming weeks, regular watering is required, at least 3 times per week. After approximately 6 to 8 weeks, the survival rate of transplanted rooted cuttings was about 85%. In another way to look at it, for every 100 cuttings that I planted, approximately 65 to 70 plants were produced. Leptospermum continentale was also selected as a potential species for plantations in South Australia. Although producing the lowest DHA levels compared to the other 2 South Australian species, it can be found in a greater range of environments. It also produces similar or greater levels of DHA compared to Leptospermum scapparium, and therefore is likely a good contender for plantation establishment in South Australia. As for scapparium, I investigated the effect of genotype and auxin application on propagation and transplant survival. I also investigated when adventitious roots were being formed on these cuttings over a 12-week period to identify the best time to transplant cuttings. Finally, I looked at how to improve transplant survival rates. The material for this experiment was collected from a wild population in the Adelaide Hills. It was then transported to the University of Adelaide for preparation. Over 2,500 semi-hardwood cuttings were planted to investigate the propagation of Leptospermum continentale. The results from this experiment suggest genotype has an important role to play in the propagation capacity of Leptospermum continentale. However, this is also influenced by the fact these plants were collected from the wild, where nutrition and irrigation is not at optimal levels for the production of material for cutting propagation. This research suggests that cuttings should be transplanted at 9 weeks after the propagation event. This is 3 weeks longer when compared with Scoparium, which is transplanted at 6 weeks from the initial propagation event. It is also advisable that cuttings are hardened off prior to transplanting and planted into a good quality potting mix that meets the Australian standards for premium quality. In summary, Leptospermum is an important genus for bioactive honey production. It can be propagated from either seed or clonally. Given the right environment, Leptospermum continentale and Scoparium is readily propagated from cuttings. Leptospermum scoparium can also be successfully cloned from wild populations. The economics of plantation establishment using seeds or cuttings needs further research. The economics will depend on the benefits gained from clonal plants compared to seedling-grown plants. I would like to acknowledge the CRC for Honeybee Products, Spring Gully Foods, the University of Adelaide, and my supervisors for the support that they have given over the course of my PhD. I would also like to give a special thank you to my family and friends who have supported me throughout my PhD. Thank you.